So first up is John Tuthill. He is an assistant professor at University of Washington, and he's going to give a brief talk titled How Flies Move Their Legs. John will be followed by Andrew Miri, who is an assistant professor at Northwestern, and Andrew will talk about neural mechanisms of movement execution. And then finally, we have Eva Dyer, an assistant professor in the Department of Biomedical Engineering at Georgia Tech and Emory. And her talk is titled The Integration of Connectivity and Gene Expression Datasets to Learn a Multi-Scale Parcellation of Thalamus. So again, we will not have Q&A, but feel free to find these people during lunch or any other time to ask them questions. Thank you.
Also, I live in Ballard, so you can feel free to just knock on my door and follow up on anything in my talk. So I'm happy to give you an update on what we've been up to in my lab. So I started my lab just a couple of years ago. For the past year, sadly, maybe depending on your perspective, I spend most of my time staring at fruit flies running on treadmills. So my lab is interested in Drosophila locomotion, both from the sensory side in the sense of how is an animal sensing where its body is and how it's moving through space, and how is it that it then is using those signals to adapt and plan future movements. So if we slow down a fly running on a treadmill, you can begin to appreciate how virtuosic its motor control is. So here we've slowed this down about 20x. We're tracking the joint between the femur and the tibia, and then we're just plotting here the angle at that particular joint. And what you can immediately see first is that the fly is walking very quickly, so it will take between 10 and 20 steps per second with each leg. And also, if you look very carefully at both the movie and the kind of statistics of how it's walking, what you see is that its locomotion is not completely stereotyped. These are not just little robots. They're able to sense and adapt to their environment. They can plan to step over objects, learn to step over objects, and navigate through rugged terrain. And so my lab is really interested in how it is that the animal's nervous system controls this behavior, and how it does this specifically with a very tiny nervous system. So this is a Drosophila central brain and ventral nerve cord, which is primarily what my lab studies. So the ventral nerve cord, or VNC, is analogous to the spinal cord in vertebrates. It has about 20 or 30,000 neurons, and it's connected to the central brain, which has about 130,000 neurons by about 5,000 ascending and descending axons. So here we have an extremely small, about the size of a sesame seed. The central brain and the ventral nerve cord both are, and relatively compact circuits that are controlling pretty remarkable behaviors like walking. And we have genetic tools in the fly that allow us to label, manipulate particular cell types in the nervous system. So my lab is really interested in starting at the periphery and both the motor side and the sensory side, and looking at the circuits that connect proprioceptors to motor control centers that are comprised of interneurons and then projection neurons, for example, between segments and up to the brain, that mediate locomotion and proprioceptive sensing. So when I started my lab a couple of years ago, we started by looking at a population of leg proprioceptors, 133 cells that are located in the fly's femur, and they're connected to the tibia by a tendon. So when the tibia moves relative to the femur, it pulls in the dendrites of these neurons. And the axons of these neurons then project into the fly's ventral nerve cord. So here the ventral nerve cord is organized, you can see, into 6 compartments that correspond to the 6 legs. So these are the axons that are coming in from these proprioceptors in the fly's front leg. And a postdoc in my lab, Akira Mamiya, developed a system that allowed us to take control of the fly's leg movements and essentially deliver proprioceptive stimuli to the fly while we record calcium signals from their axons. So what Akira does is, or what he did, was to glue a tiny little steel pin onto the fly's tibia and then use a magnet mounted on a high-speed servo motor to move the fly's tibia relative to its femur while he would track with high-speed video the angle of the femur-tibia joint. It looks something like this. So this is compatible with 2-photon imaging. You can see the laser scanning here, and Akira is just moving the fly's tibia, and the angle between the femur and the tibia joint is plotted here. So Akira used this system to investigate how different subtypes of leg proprioceptors encode movement at this joint. And so I'll summarize an entire paper in just one slide and show you that what he found is a population of proprioceptors that we call the claw, based on the shape of their axons, that are tuned very specifically to the position of the leg, in the sense that when the leg is flexed, so here in this plot the leg starts extended, flexes, then goes back to extension, you see that some of these axons light up just when the leg's extended, and other ones light up just when the leg is flexed. And you can see that in this little cartoon here, where this shows the angle of the femur tibia joint, and then this is the change in calcium signal within these axons. He then identified a second population of neurons that are responsive only to the movement of the leg, and they're responsive in a bidirectional way, where if you start off with the leg extended and flex it, and then extend it again, it responds to each one of these movements. So these are bidirectional movement sensors. And then finally, he identified populations of directionally sensitive neurons that respond only when the leg is, for example, moving from extension to flexion and not at all in the other direction. So we think of this as kind of the basic peripheral sensory code for how the fly knows where its legs are and how they're moving through space. And now what we're doing in my lab is moving from the proprioceptive system into the central nervous system, looking at populations of interneurons, intersegmental neurons, and in particular motor neurons that are controlling locomotion. And so in the next few slides, I'll just give you kind of one slide updates of exciting directions that we're headed in. So the first of these is, again, fly walking on a ball, tethered. We are now expressing channel rhodopsin, specifically crimson, and the fly's leg proprioceptors. And we are able to, by focusing a laser, particularly on one leg, able to manipulate activity in the fly's proprioceptive system for just a few milliseconds and look at the effects of behavior. So while we do these experiments, we're tracking the legs in 3D, and we're asking, what is proprioceptive feedback providing to the nervous system to allow it to plan its future movements? Another postdoc in the lab is looking at motor control, specifically how individual motor neurons contribute to movements of the legs. So this is just an example where he's patched onto a motor neuron that controls flexion of the tibia and then is driving activity in it and looking at how it controls tibia or femur tibia joint movement. And he's doing this comprehensively across all the motor neurons to look at relationships between function and anatomy in these populations. And then another postdoc in the lab has developed methods that allow us to record from all the motor neurons simultaneously in a walking fly. So here again, we're using 2-photon imaging. We express GCamp in all the motor neurons, the fly's walking, and we're using volumetric imaging to record from all of these motor neurons, 50 per leg, as the fly's walking. And this experiment actually provides the input to a collaborative project we have with Wei-Chung Lee's lab at Harvard, where we do motor neuron imaging, we dissect out the fly's ventral nerve cord, we ship it to Wei's lab, and a graduate student, Jasper, images the VNC. And so this is actually a VNC that we shipped to Jasper and has now been fully imaged. We have a full volume, and we're starting to identify and reconstruct particular motor neurons. This is the same leg motor neuron that I showed you a whole set of recording from. Not the same one, the same type in a different fly that we've identified in the EM volume. And our goal is to kind of reconstruct all the circuitry between the proprioceptors and the motor neurons and figure out how this whole system is put together and to generate hypotheses about how it works. And so this is my lab, and I can't take any questions, but like I said, I live in Ballard, so just come find me. Thanks. Use this one, I think.
So I don't see the monitors at all. Oh, that looks good. But there's still nothing on the right monitor. Oh, tap it. It's not there. Oh, here we go. Oh, cool. Sorry for the delay. Well, it's really nice to be back and see many of you again and get excited again about all the amazing work that's going on here. And thanks for the chance to present a little bit about my work. So I'm interested in the neural basis of mammalian movement. Amazing complexity and agility of mammalian movement is really critical to the evolutionary success of many species. And I think this is really well illustrated by the acrobatic feeding behavior you see here in the harvest mouse. But success in domestic environments can also require sophisticated motor behaviors like this one here, this cat sneak attack. Sneak attack, yeah, right there. But unfortunately, our mechanistic understanding here remains very limited. We simply don't have very good ideas about how neural activity is bouncing around the motor system in real time to generate movements like this. So the work in my lab uses the mouse, which is relatively new as a model for motor control, to answer basic questions about how the motor system gives rise to movement. So with my 5 minutes, I'm just going to tell you about the project that I'm working on, which is furthest along at this point, that we're starting to write up. Here, our graduate student, Claire Werner, has been working with me using the mouse, examining how the motor cortex helps enable the simultaneous co-contraction of antagonist muscles. And here she's using the mouse to really build upon some ideas that have come from previous work in cats and primates. So we know that the rotation of joints requires the alternating activation of antagonist muscles in order to rotate that joint. There's, of course, like the triceps and the biceps that help rotate and extend, flex and extend the elbow. But it's the simultaneous co-contraction of antagonist muscles that's necessary to stabilize joints against rotation. And this is important during many movements. For example, when we're trying to very precisely position our joint angles, to very precisely position our limb, like in the cat here when it's planning its sneak attack. In thinking about underlying mechanism here, one idea from the literature is that the cortical output driving antagonist flexor and extensor muscles is simply combined together in order to enable co-contraction. But there's also evidence that cortical output does something unique to facilitate co-contraction. There are intrinsic spinal circuits that help ensure alternation during joint rotation, antagonist muscle alternation joint rotation, like the classical 1A inhibitory interneuron. And there's some evidence actually from human studies that imply that cortical influence mutes this inhibition to facilitate co-contraction. Apparently by activating presynaptic inhibitory interneurons that inhibit sensory afferent terminals, and by inhibiting the 1A interneurons. Using the mouse, Claire has begun to dig into and test some of these ideas. So the first thing Claire did was develop a behavioral paradigm in which mice will alternately do an alternation behavior and a behavior that requires co-contraction in order to get rewards. So in one of the task phases, the animal has to iteratively turn a small wheel with its right paw in order to get rewards. And in the other task phase, the animal holds a stationary handle, and then rewards are dispensed when it simultaneously co-contracts its biceps and triceps muscle. And in each behavioral session, there's an epoch of the co-contraction behavior that's flanked by two separate alternation epochs to allow us to control for changes across time. Here you can see average muscle activity of the muscles that Claire records from in this behavioral paradigm, but our focus here is really on the biceps and the triceps that you see here in color. And in activating forelimb motor cortex with the injection of mucimol causes a dramatic reduction in task performance for both of the phases of the task. So now if pathways activating flexors and extensor muscles are just combined or co-activated during co-contraction, then we'd expect to see similar patterns or similar correlated patterns of flexor and extensor related activity in motor cortex during alternation and co-contraction. But this is actually not what we see at all. So Claire used silicon probes to record from populations of neurons in the deep output layers of motor cortex. And then she used behavioral triggered averages of the activity of all of these neurons to fit the averaged activity of simultaneously recorded muscles. And she found that models fit to the muscles during alternation, or the first alternation epoch, generalize well to the second alternation epoch, but actually do not fit data from co-contraction well at all. And she can go the other direction and fit models with the data from co-contraction. These don't generalize to alternation well at all either. We can fit these models using either all the recorded neurons or just the subset of neurons that have relatively wide waveforms, which should be predominantly pyramidal neurons. And in both cases, we see the same sort of fit quality, quantified here in terms of the R-squared values. Now, the key observation here, though, is that the neural activity subspaces inhabited during alternation and co-contraction are quite different. So if descending pathways activating flexors and extensors are simply co-activated during co-contraction, we'd expect the same subspace to be inhabited during these two tasks, but that's also not what we see. So the overlap In the activity subspaces for the 2 alternation epochs is about 50%, so there is some change across time. But the overlap we see when we compare the activity subspaces from alternation and co-contraction is much lower. It's down around 20%. And that 20% value is actually on par to the amount of overlap we see in the subspaces inhabited during alternation and when the muscles are actually completely inactive. Now, to show the activity subspace differences we see here hold for specifically the activity which is muscle-related, we projected all the neural activity down into a subspace defined by those linear models that I told you about in the previous slide. So each of those linear models basically just defines a direction through the full neural activity space. Collectively, then, the models fit to several different muscles, define a low-dimensional subspace, and when we project all the motor cortical activity through that subspace and do the same sort of analysis, we see the same sort of trend here. And in fact, this trend also holds for activity specifically in corticospinal neurons, which are the relevant output neurons here. We made GCAMP6F fluorescence measurements from the apical trunk dendrites of corticospinal neurons to estimate their calcium fluctuations and spiking. And we see the same differences between activity subspace overlap in terms of the calcium and the spiking for these cells specifically. So collectively, these data seem to indicate that motor cortical output is engaging spinal circuits in a unique way to enable co-contraction. And we're currently analyzing similar data to test this prediction that motor cortical output specifically activates presynaptic inhibition during co-contraction. And here we're using our transsynaptic rabies approach to measure activity specifically in the subpopulation of corticospinal neurons that synapse directly onto the presynaptic inhibitory interneurons. With that, I'll just end quickly, say thanks to all the people that helped here, including Claire Werner, who did all of the beautiful work. And thanks again for the chance to present. Thanks.
Hello, excited to be back here again today to talk about some updates from last year. So today I'll be talking about a project which is in collaboration with Julie Harris here at the Institute to integrate different types of anatomical data sets in order to learn parcellations of the brain automatically. And then at the end, I'll talk a little bit more about another project that I'm excited about. So just sort of the overarching aims of my lab, we're interested in being able to develop automated techniques to take images and translate those into comparative models that tell us things about the brain and how it changes. And so in order to do this, we're interested in a few kind of key main outputs. So in order to analyze and compare these brain images, we are developing compact abstractions of brain structures, so things that allow us to take complex data sets, images, and boil them down into a simplified representation that we can then compare and contrast. In order to do this, we also need methods to then extract relevant features from images directly. And then finally, building new methods and models for learning variability across many different samples. And we're using that as a way to study inter-subject variability, so looking at changes across subjects, across different brain areas, and also looking at different time points in aging and disease. And so what I'll talk to you about today is some work that we've been doing to follow up actually from my last year's talk. I did talk about this, and we're sort of making some progress in integrating other types of information into this problem. So our goal is to really develop automated techniques for doing parcellation of brains. We've been focusing to start on the thalamus, but this is an idea that we really want to expand out into whole brain data sets eventually. And so really our goal is to take 3 different types of data. We're looking at the mouse connectivity atlas. So this is information about projections that are coming from many different brain areas. So a virus is injected into a particular brain area, and this will then produce some signature in a downstream voxel, right, or a spatial position within the brain. And so in addition to these connectivity data sets, so using projections, we're also now looking at cytoarchitecture, so how are cells packed and how is this different across different areas, as well as whole brain gene expression coming from different Cre lines here developed using the same actual imaging methodology boundaries across all three of these different cases. And so the basic idea is to basically zoom into a particular spatial position in the brain and look at the changes in either the cytoarchitectural patterns, the gene expression, or the different projections that are coming from these different injection sites. And then our goal is to eventually be able to predict the different areas in the brain, hopefully to eventually resemble the most recent common coordinate framework that has been developed using these different sources of information here at the Allen Institute. And we want to be able to do this all automatically. And so what I talked about last year was a sort of, I guess you could call it a naive approach to do this. And so we've realized why it's naive and how we might be able to improve upon it. So to start, we're looking at many different projections. So this is about 3,000 different projections into different brain areas, as well as about 60 different Cree lines. And so we can build up a representation of each of these voxels in terms of all these different patterns or signatures of the anatomy. And then to start, we were just interested in kind of going at it and applying different clustering algorithms to see how well we can actually parcellate the brain using this approach. And so what we find here, this is just a rendering of the result from using a k-means clustering algorithm when we combine both genes and connectivity versus genes alone. What we find, so here on the left is actually, oh, and I should say that this is focusing in on thalamus, and so we're zooming in here just on thalamus in this parcellation. So on the left is actually the CCF, and on the right is the result of our clustering algorithm for these two different cases. And the ARI is a measure of how well the automated clustering actually matches the CCF that was developed here. And so what we find is that there are some structures that do seem to be, kind of pulled out using these sorts of approaches, but we were interested in how we can actually push this further and use information coming from these different data sets in a more parsimonious way. And so in order to do that, we developed a number of techniques to basically do some dimensionality reduction, which allows us to then find simplified factors that kind of combine genes and connectivity into one common representation, and then from that, then cluster those multi-modal features. And so basically we take our data, as I showed in the previous slide, and our goal is to basically factorize it into two parts. We've played around with a lot of different types of dimensionality reduction techniques. We've already heard a lot about PCA and some of these other techniques, but what we've actually found has been really useful for solving this problem is to actually use something called dictionary learning. It's a form of doing sparse matrix factorization, and really the goal is just to split this matrix A into two parts, and the goal is to basically find coefficients V that are sparse. And so the idea is basically you learn these dictionary elements or these factors of this multimodal data that then give you a sparse representation of the rest of the data set. So we used this particular technique, and what we found is that using these sparse matrix factorization techniques really do give us a much better agreement with the CCF. And so we're starting to dig into the results of this to really understand when and how these sorts of techniques can give us a useful parcellation of the brain. And so I'm just pointing out a few structures here that we found with these new revised techniques. We can actually get much better and clean agreement with the CCF. And so in addition, we're also exploring ways of doing this in a supervised manner and to really understand how exactly trained neuroanatomists go about defining different boundaries. And so this is just an example of a supervised learning algorithm where we basically ask the system to predict which of these different projection or gene expression data sets are most representative or can predict a given brain area of interest. And so this is just an example of showing these coefficients that pop out and the underlying experiments that are predictive of, in this case, LGD. And so we can do this for all the different structures in thalamus, and we're starting to dig into that with Julie's team as well. In addition, we've also been developing methods for parcellating the brain and finding layers using cellular densities. And so this is just an example of some tools that we've developed for cytoarchitecture estimation. Here I'm just showing results on a retinal sample where the layers are very easy to determine, and then we're also now applying this to the Allen Reference Atlas, where we can actually pull out, in an automated way, cellular density counts, so we're getting individual cell counts, as well as then looking at how our estimates of the different layers in cortex overlay with the reference atlas as well. And so we're scaling this up currently, and we're excited to then incorporate these cytoarchitectonic maps into our multi-modal parcellation strategies that I described before. We're also applying these same sorts of techniques now to three-dimensional data sets. So this is a X-ray micro-CT data set where we've already pulled out all the individual cells across a long stretch, going from hypothalamus all the way through thalamus, the reticular, or the TRN and VP, all the way up into primary somatosensory cortex. And so we're using these same change point detection strategies that we've been using for finding layers to now find individual brain areas and then automatically analyze the underlying anatomy in these areas that we've discovered. And also we can see nice layers and even barrels within somatosensory cortex with this technique. And so in terms of the next steps along these two different aims, we're interested in incorporating, as I said, cytoarchitecture data, looking at ways to both factorize the data using multimodal and also unimodal features as well, and characterizing gradients and smooth variation in the gene expression, which we've seen in these data sets thus far, and using this idea of cytoarchitecture estimation to now study retinal degeneration and other changes in the layers and cell densities that occur in disease. And the other thing that's happened over the past year is that we've expanded significantly. So this was a little bit after I was here last year, and now this was my lab at our recent retreat. So we have a really great team of people, and I'm really excited to be here to tell you all about the work that we've been doing. So I think, one of the things that has come about over the past year is after becoming an NGL, I was able to meet Julie and a lot of other great people here at the Institute. And this has really kickstarted my career, and it's really created a very fruitful collaboration. And so I'm excited to be a part of the family now. So most of the work on the multimodal parcellation has been done by my student, Eric Jorgensen. And I'd also like to thank my funding sources. Thanks so much.